Morning, everybody. Welcome to day 2 of the media meeting of the Indian Academy of Sciences. My name is Narendra Dikshit. I'm co-organizer of this. It seems on. Maybe I need to get it closer to me. Okay. Am I audible now? Yeah, okay. Yeah, so welcome again. I'll just finish. It's fine. Yeah, yeah. So my name is Narendra Dikshit. I'm a co-organizer of this symposium on AI in health, together with Professor Somdatta Sinha, who's sitting here in the front row. First of all, we'd like to thank the Academy for letting us host this symposium. This is really Professor Somdatta's brainchild. I've been playing side fiddle, perhaps, in helping her put this program together. What I'm going to do is in the next 5 minutes or so, set the stage up, give you a preamble of what this whole symposium is about, why we are trying to put it together, and where this might take us going forward. So whether we like it or not, I'm sure everybody is realizing that we are in the AI era, and AI is touching lives in many different ways. And one big area in which it's having an impact is health. It's ceased to be a tool for research alone. It's making headway in a way that it's beginning to impact clinical decisions. So when that happens, it becomes important for us to see whether we're able to take stock of where this whole thing is currently and where it's headed. And as you will see, because the whole thing is so promising, there are people in academia, people in the industry, big and small, startups, large industry, as well as people in the government that become involved. And the government's involved because regulatory principles have to be established for these new protocols that are coming our way. So we thought this would be a good forum for us to put together a set of talks that would give us a perspective on each of these different directions in which AI is headed as far as health is concerned. So that's the basis behind the set of speakers that have been invited to give us their take. They are all leading luminaries in their own disciplines to tell us about where this field is headed in these different directions. Of course, from the program that you have circulated, you'll see there's a bit of a change. One of our speakers, Professor Nishit Srivastava from IIT Kanpur, has had to cancel his trip at the last minute because of a personal emergency. And so that part of the program will unfortunately not be represented. He was supposed to talk about the government's initiative to build reliable AI systems and to test the reliability of new protocols that people could construct. And so that arm we'll have to sort of think about separately. But the rest of the speakers will touch upon the other aspects of where AI is headed with respect to health in the next 2 hours. So let me briefly introduce to you the speakers. I'm going to take a few minutes to say this now so that we don't spend time introducing them between their talks. Our first speaker will be Professor Sushmita Mitra. She is a professor at ISI, Kolkata. She was formerly the head of their machine intelligence unit, where she is also currently professor. Her work has been at the intersection of data sciences, algorithms, and healthcare, particularly focusing on image processing. So she's going to tell us about the many different algorithms that their group is building that can look at imaging data of very different kinds, X-rays, CTs, and so on, and help in decision making for clinicians. So what she calls, and what I think the field also calls, assistive intelligence. So we'll hear from her on that front. She's an accomplished researcher. She's, of course, fellow of this academy, fellow of many other academies, and she's also a J.C. Bose fellow at ISI. Our second speaker is going to be Dr. Geeta Manjunath. She's an alumnus of this institute. She is founder and CEO of startup company Niram AI, and they have put together a product. So this is an example of where AI has helped build a real product where they can use thermoscans of skin tissues and tell whether somebody is likely to have breast cancer. So this is an early screening tool that combines existing knowledge of biology, and using tools from AI, they have a product. And this has been used to screen many lakhs of women across more than 20 countries. She has many US patents to her credit. She has, of course, a PhD in artificial intelligence from our institute and a management degree from Kellogg's in the US. And she was named in 2020 as one of the 20 self-made women in India, right? So we're going to hear from her about how AI has been used to construct or build a translated product. At this point, we'll switch gears. The first two talks will tell us how AI is helping us piece together existing biological knowledge to make headway with diagnosis, treatment, and so on. In the next two talks, we'll see if AI is able to help us discover new biology. So the third speaker, Professor Nagasuma Chandra, she's of course a faculty member here, a dear colleague of ours. She'll tell us about using AI tools for analyzing different types of multi-omics data and understanding new things about disease. So she is, of course, here a professor of biochemistry. She's an expert at computational biology, systems biology. She's been developing these sorts of algorithms for many years now. And today she'll tell us how some of these algorithms can help us think about what sort of immune responses predominate different types of disease pathologies, and how they can help us drive interventions that are more focused, more targeted, and therefore more impactful. She's also a fellow of this academy and all of the other science academies. And she also is a founder of a new startup called HealthSeal. So she'll tell us a little bit about that startup also, perhaps. And our last speaker is Dr. Oishi Mazumdar. She is from TCS Research in Kolkata. She has a degree in automation and control, and she's been developing, she's been part of the TCS's efforts in developing what they call digital twins. So here we are going beyond just thinking about a particular disease case. They want to see if an entire organ, or perhaps an entire system of organs, a whole body perhaps, can be built digitally in a way that is so powerful that you can ask what if questions. What if I do something to this? What's likely to happen? So this is going to accelerate drug development, discovery of new pathologies, and so on. And the hope here is that these AI tools help us build platforms that are more rigorous than what has been possible using simple mechanistic modeling approaches. She's, of course, highly accomplished in her own space. She has many dozens of patents and has received many awards within TCS as well. So that's the broad program. So you'll see as we go through that we start out with AI algorithms that help us piece together existing biology to come up with smarter decisions. There are products that startup companies have made that are deployed. There's new biology that can be discovered and that can be scaled up to a point where disease interventions can become much more logical and rapid. And finally, Professor Somdatta Sinha will give us closing remarks to tell us where this whole thing is headed. So logistics-wise, each speaker will have 20 minutes to talk, and that will be followed by roughly 5 minutes of question answers. So with that, I'll stop and invite our first speaker, Professor Sushmita Mitra, to give her talk.
I put it here. And the slides with this. Okay. A very good morning to everybody. It's my great honor and pleasure to have been invited here to deliver this talk. I thank Professor Sinha and Professor Dixit for giving me this opportunity. So I'll be giving you a brief overview of the research that we've been undertaking in our group for the last several years. So we've been working on machine learning for long and deep learning thereafter. So to give you a brief introduction, when you have machine learning, the traditional kind of learning, you have to have a domain expert to extract handcrafted features, and then you feed it to a decision-making system. However, in deep learning, you have huge amounts of data as is available today in multimodal forms, and then the system on its own extracts the features and arrives at a decision. So shallow and deep learning, as I've said, so machine learning traditional is shallow learning. You don't have so much data. You have a domain expert to extract the features, whereas it is otherwise in deep learning. So the key advantage of deep learning is in automatically learning good features. So why in medicine? Because there's a lot of imperfection, imperfect knowledge, there's incomplete understanding of the biological mechanisms, imprecise test measurements, uncertainty of the ranges of the results, simultaneous presence of more conditions, more than one condition, missing information. Therefore, you have the scope of learning, some kind of intelligent processing and automated analysis, and now it is as assistive intelligence and not as a self-sufficient system, because we don't intend to replace the physician or the surgeon, but to assist them given the huge volume of data, because it is the human end user who is responsible for any medical decision. And also you need interactive or active learning, because there is insufficient annotation, and thereby there needs to be a continuous feedback between the medical professional and the system. Then there is a genomic issue to that. Of course, we'll be hearing about that in one of the subsequent talks. So it is found that there's a domain called personalized medicine, which needs to offer the right drug to the right disease at the right time with the right dosage. So given a set of patients and a certain drug prescribed, depending on the genetic composition of the individual, you can have 4 different situations. And we are obviously interested in the situation where the drug is neither toxic but is beneficial. So the genomic component also has a major role to play. Today in the hospitals, you've got robotic surgery. Recently in Eastern India, Manipal hospitals projected this, and so you have Da Vinci, the robot. Then India made SSI Mantra, which is India's first indigenous surgical robotic system at half the cost. And the advantage is that since there is precision in the measurements, you use the medical imaging, and the surgeon looks at the machine, whatever the robot is seeing, and operates the robotic arms to get a high precision surgery, which results in a faster recovery and loss of fewer working days of the active person. The patient can get back to life, a normal life. So this is what is segmentation. When you have a, let's say this is a brain scan. So there is a tumor there, brain tumor, the inner part. And when the system can give you an automated delineation of the different regions, it can act as an assistance to the surgeon, who can then take the decision and operate. So in our team, we have been working on these aspects like automated severity level grading of lung diseases, automated grading of prostate cancer, early screening for diabetic retinopathy, then segmentation with resource efficient models, because resource efficient means lightweight models such that the carbon footprint is less, because of course you have the computational expenses involved, and we don't want to make it bad for the environment, as good as possible. Because each GPU consumes a lot of computational resources. So then disease classification in multimodal framework and computer-aided diagnosis with imbalanced data. Why imbalanced data? Because in medical images, in disease images. Imbalance is a major issue because suppose you have a tumor in a certain slice of the brain, then obviously the tumor constitutes a very small percentage, let's say 0.5% of the voxels, whereas the majority of the region is consumed by the background region. So if you want to have a decision, and if you are unable to pinpoint in that small tumor area, even then your model will give you 99.5% accuracy, which is meaningless. So you need to focus on the low volume, or let's say those 0.5% of actual region which is of interest, the disease region. You want to operate that. So this is called imbalance learning. So you need to develop certain loss functions, algorithms to handle that. So this is another important problem. Histopathology. First, let's go to histopathology. This is, of course, you know that in cancer diagnosis, this is the biopsy is the gold standard even today. So you have to look at the scanned tissue slides, and the pathologist looks at it and makes a decision. And since there is huge volume of data, automated processing has a great possibility. Now, that is not a simple problem, which we'll see just now, because there are different issues involved. So this is a low resolution image of the slide. This is a medium resolution. So this part is expanded here, and this part is expanded. This is the high resolution one. Obviously, the high resolution image gives you more detail for the doctor to see. But the caveat is that the computational complexity will become much larger, because here the resolution is higher and the number of resources you need is much more. So we need to arrive at an offset, and it is a truth, it is a fact that academia doesn't have that much computational resources compared to industry. So we have to arrive at an offset. So when we did the experiments on this kind of data, we were forced to stick to the medium resolution one. Then the other aspects are like image blurring, then tissue tearing, tissue folding, and staining from a marker. So these are difficult for a computer to analyze. Maybe a human being can handle it better because of the better cognitive capabilities. Then when the tissues are coming from different pathological labs, the stains used can have different shades. And then the same tissue from the different labs can have this kind of variations, which obviously becomes very difficult for a machine to analyze. With that, let's move on to the first application that I start to explain here. So the whole slide image of prostate biopsy, of course, this was taken from the publicly available PANDA challenge on Kaggle. And what is normally done is a Gleason pattern score is provided. And today you have the ISUP grade, and the transformation from the Gleason to the ISUP grade is according to this table. And what happens is the majority, the highest grade obtained in Gleason score and the second highest grade obtained in the Gleason score, they are summed together by this tabulation. And ISUP grading is supposed to be a standard one today in prostate cancer grading. So what we did was, so the whole slide image you see is so large, a CNN or a standard deep model cannot handle such a huge volume at the input. So we had to resize. Now resizing destroys details. So we extracted some small patches from each of the slides and discarded the patches that were mostly white. Now the problem that remained was, for the whole slide we had the grade because it was annotated. We didn't have the individual grades of the patches. Okay, so this was the model that was used. Of course, we used the EfficientNet B1 model because it was lightweight. We needed less resources. So this was the whole slide. We partitioned it into bag of patches, and we had a hierarchical framework of the EfficientNet, because it was observed that the grades 0, 1, 2 were more clustered together, had more confusion within them, and the grades 3, 4, 5 were more clustered, because obviously 3, 4, 5 were the higher grades and 0 is the normal, no cancer, and 1, 2 is the lower grade. So anyway, this is the structure. Uh, uh, the next one was on COVID-19, also on, this was done quite a few years back during the COVID times. We were sitting at home, and this is the publicly available data. You remember it first came to China and not much data was available in the public domain. So this was how a lung looked like in those times. And so at that time, there was not much annotated data available. So we had the training data, which had only 2 classes because annotation was insufficient, and the testing data had 4 classes. So this is the beauty of the problem, because you're training on the data set A, the sources are listed here, and the testing was made on the superset of the training set in that sense. So we propose the domain extension transfer learning, where the domain D is represented by the tuple where this domain is the domain DS is a subset of the target domain. So the objective of transfer learning here was to learn the target conditional probability distribution in DT with the knowledge gained from DS by solving TS such that DS was a subset of DT. So in the model form, the CNN was trained from scratch on the 2-class problem. The final layer had 2 nodes. And then we replaced the last fully connected layer of the pre-trained CNN with 4 nodes, because the target domain had 4 classes, and the rest of the weights were frozen, and only the final layer was trained, and the evaluation was on the data set B, which is a superset of the training set. We use 3 conventional well-known models, AlexNet, VGGNet, and ResNet, with this domain extension transfer learning, and the confusion matrix showed that it worked pretty well on the test data. This was a visualization generated with the GradCam. Next, we move to the diabetic retinopathy problem, which we handled thereafter. This was a project we had actually under DBT with Shankara Netralaya and Mohan's Diabetics in Chennai. So this is these basic slides are borrowed from my medical collaborators. So diabetic retinopathy, you know that India is the diabetes capital of the world, and diabetic retinopathy is a disease which silently affects the eye of a huge number of individuals, particularly in the village areas, because you know the ladies don't go to the doctors often, and they think it is cataract or eyesight myopia. And when they realize what has happened, by that time it is irreversibly gone, and it's causing blindness. So this is the non-proliferative or the preliminary stage, the different stages, and then this is the proliferative or the severe stage, where it is a point of no return. The objective is to catch them, screen them, so that you get it at as early a stage as possible. And the tragedy is that 90% of the vision loss due to diabetic retinopathy is preventable if it is detected early and treated timely. With that in mind, you need to screen them regularly. And the problem is that we have 22,000 ophthalmologists in India, but over 74 million people with diabetes, particularly in the villages, to be screened annually. And in diabetes, what happens is there are different kinds of pathologies which develop in the retina. And these are microaneurysm, hemorrhage, soft exudate, hard exudates. And the doctor grades the diabetic retinopathy by, if you divide the eye into 4 quadrants, so how many of the quadrants are affected and to what density they are affected, that determines the grade. In this project, up to now, we have been able to generate. Now, Indian database creation is a very important problem. As you know, in the publicly available domains, you have mostly the Caucasian data. And even if Asian, it's the Chinese data. Getting Indian data is very difficult. And we know that the genomic-wise, we are different, and training on the other kind of data and testing on Indian data becomes a major issue. So one of the major problems is generating Indian data. So here we generated a large-scale, richly annotated, multimodal diabetic retinopathy database, SANMOD, from Sankara Nethralaya and Mohan's Diabetics. And this was the data set reported, and thereafter we benchmarked it, and this has been recently published. So it provides 4,200 high-resolution fundus images, and it has the different kinds of pathologies. And after that, we have been developing a tool for this. It looks like this. We won't go into the details because I think we'll be short of time. So these are the different kinds of pathologies that stand there. And I'll just skip through these because I don't think we have time. So this is weighted deformable DeepNet. So we introduced, so this is a PhD student who has finished the PhD, and she introduced the deformable convolution. So the deformable convolution is able to handle in a better manner for segmentation. So this is the normal convolution, and this is the deformable convolution. So we are able to model in a better manner. This is the architecture and the comparison with state-of-the-art models. This is how over the iterations, the modeling becomes improved when you have this deformable convolution. This is the parameter-wise contribution that ours was computationally competitive compared to some of the others. And finally, the hybrid models. This was also the contribution of one of my recent PhD scholars who is present here today. So you see what, so the convolution network have a complexity order of n, vision transformer have order of n square, the state space and XLSTM have order of n. So here we have 4 kinds of hybrid models: the Wave Coformer, UVix, LSTM, ROTUVL, and EcoMamba all pursue global understanding at linear cost. So in Wave Coformer, you have a wavelet component to handle the temporal or the spectral features along with the spatial features. This is the structure of the model and some qualitative map of how it improves. It reduces the false positive and the false negative and increases the true positive at a reasonable computational cost. Then the Vision XLSTM, to overcome the high computational burden and the high resource demands, we explored the Vision XLSTM to capture the global context in a computationally efficient manner. So you have the architecture like here you have the Vision XLSTM, the bottleneck. Thereafter so this is the qualitative map. Thereafter we introduced the rotational model, the rotational UVIX LSTM to handle the volumetric aspect from the different angles. So it was modeled like here you had the rotational model so that the volumetric information could be better represented. This is the qualitative map. And finally, the state space model, which had the sequential bottleneck had to be removed. So the EcoMamba net with the complexity order of n. And this is the architecture. So you have the module here. This is how the qualitative map looks like. And so now the comparison between these 4 hybrid models based on the number of parameters, the number of T-flops, on the time and the memory. And now when to use which of these 4 hybrid models. So when you need maximum accuracy on edge or mobile devices, we propose ROTUVIL, UVIX LSTM for real-time applications as in image-guided surgery. It is fastest. And Wave Coformer for offline analysis as its spectral spatial fusion achieves top-tier overlap accuracy. EcoMamba net for accelerating clinical workflows on limited clinical hardware. And now I won't go into the detail, but the thing is that misuse of AI can happen when you just ask chatbot everything. And then I thank my students, some of my students without whom this journey would not have been possible. Thank you. Any questions?
So nice talk, ma'am. I just wanted to know from your model of AI, how accurately did you predict the COVID pneumonias? I mean, you had those 4 different divisions.
At that time,
At that time.
yeah. So
How
at that time, the accuracy was good, but you see, the limited data set available was not very great at that time. And the more data you have more accuracy, higher accuracy. Deep learning needs more data. At that point of time, it was good. But obviously it will be better if it is done on present day data.
So I am a clinician. First of all, it's surprising that we have a session on AI in medicine with not a single speaker from medicine. People whom we are trying to assist. That's interesting. But let's go away from that. Our understanding on the clinical side, people who have been seeing patients, and there are systematic reviews showing that the performance of AI in diagnosing and helping with diagnosis of COVID-19 was absolutely zero. The problem is, there are multiple problems. The first thing is, if you ask 2 doctors to look at the same thing, they themselves don't agree. So we are actually trying to predict something which is, we don't know what we are trying to predict. Secondly, the biggest problem that I see as a clinician, it's a black box. When you take 1,000 COVID-19 X-rays and 1,000 non-COVID X-rays and you use AI to distinguish and find some features, till I don't know what it is looking at and whether that's a good predictor, I am not going to use this. And that is, I think the basic thing is believability and the liability. And the thing that I'm worried about as an elder physician is that when AI shows me something and when I look at it and I find that there is something odd, I'm able to call it out. My problem is my younger colleagues who are now trying to do this on AI, they look at it and they don't even pick up the difference, and they don't pick out we somehow think that everything in medicine is science. Unfortunately, medicine is far. When I see something and there is something unusual in a corner, I raise an alarm and my brain thinks that there is something different. The biggest problem is when AI tells us something, we are not willing to believe it. If I were to do today and it were to tell me that Jupiter is the smallest planet and AI were to tell me, I would believe it. That's where the problem with medicine. Yeah, that's the question. Yeah.
Sir,
The question is COVID, in our understanding.
Sir, I got a part of it. So the thing is, just like you are a senior physician, and you have said that your younger colleagues take it in a different way, handle it in a different way. Similarly, you have been trained on the data or on the techniques by your seniors or by looking at the patients. Similarly, the AI model is learning from the data. It's some kind of human-like learning. And so the more data it is exposed to and the more situations it is exposed to, its learning will improve. Just like you are able to distinguish that certain thing is happening in the corner, similarly, a new model may not be able to handle, just like your younger colleagues. So given large volumes of data and of course high computational power, which is why we want to get lighter models, then we can expect that the AI model will become better. And that's why we don't intend to replace a physician, but to assist, so such that experts like you are at the end to take the final decision. And today you have interpretable AI. So it is providing some interpretation on why it arrived at a decision for people like you to verify, and whether you wish to accept it or not is on you.
Yes. So that was an excellent talk. My question was about the prostate cancer stratification that you showed. So my real question is an extension of what was being discussed earlier. To arrive at that score, how how comprehensively did you have your training data so that you could minimize the number of false positives?
So,
Because over there, the real challenge is tumor heterogeneity as well.
so that we had a doctor from the Tata Institute of Cancer Research in Kolkata in that research, but the real problem was getting the real data. We worked on the publicly available data, so that is a disadvantage to that. So given the limitations of the whatever data we were exposed to, we could validate with the doctor involved about the results that were obtained on the unknown data which was available in that set. But of course, I cannot vouch for what will happen when some other unknown data is provided, because then the training needs to be taken. So that was more an academic work. And of course, to make it deployable, it needs a more exposure. Exposure is an issue.
I think in the interest of time, we'll have to stop. But let's thank Dr. Mitra again. All the speakers are available over coffee. Thank you very much.
Thank you.
And I'll request Dr. Geeta to. So while Dr. Geeta sets her talk up, I'm just going to address the comment that our clinician friend has made. It's true, reliability is an issue. The first speaker, who unfortunately couldn't come, was supposed to speak on that. And the other comment I want to make is all of the colleagues here who are speaking, they work very closely with clinicians to make sure that what they're saying is applicable to the clinic. Thank you very much, though.
Okay. Yeah, good morning, everyone. After that wonderful sort of a broad talk touching multiple problem statements, giving an idea of how AI can really be used. I'm happy to sort of come back on stage on my favorite academic institution where I did both my master's and PhD, and where almost my journey of a startup started. Yeah, I'm Geeta, founder CEO of Niramai. Basically, I was a principal scientist at Hewlett-Packard Labs after I finished my PhD, and then I was head of AI after 17 years at Hewlett-Packard at Xerox. And of course, I was working on multiple research problems, including COVID, and like one of them, what is the impact done actually along with Chiranjeev Bhattacharya ji at CSA. We actually did develop a AI-based COVID screening model used by 10,000 doctors, in the rural areas to say whether they need to really send to a PCR or not. So some of these are possible to be deployed, especially in low resource settings. And what I'm going to do today is to sort of focus on one problem and my journey as a scientist to quit my job and become an entrepreneur, because I really wanted to see how an AI model can go on the ground and make it. And the learning is what I thought I will share, which adds a different color to what ma'am presented before. And thank you so much, Professor Sinha and Professor Dixit, for inviting me. So I think it's very obvious. We talked about how AI is changing the world, and particularly if you look at AI impact in healthcare, pretty much healthcare is the last impacted in AI. Of course, we use Uber, Ola, all of them, many other wonderful applications we have just gotten so used to. But healthcare is the least or last one, which is getting, and this symposium is right on time. And we talked about multiple use cases, and you have many speakers going to talk about individual aspects of it. The place where AI has started making an impact, even around the world, is in the analysis of medical images. As the first phase of making a clinical decision support or supporting or assisting in clinical decision support. Of course, using AI in a non-clinical setting, like matching the patient with the right doctor and prioritizing the patients for review by the doctor. Those things are a little bit more similar to the other domains. Here, when it comes to clinical decision support systems, like previous speaker mentioned, the class imbalance becomes very, very important. What do we mean by class imbalance in this scenario is that number of people who are healthy is much, much more than number of people who are unhealthy, which is good for the company. I mean, good for the world, but it's very bad for a machine learning, engineer or a researcher, unfortunately. And that is the huge imbalance problem we are trying to solve. And when I go to a small class where they're trying to say AI in healthcare, I say, see, let's say, actually, like, we can have like one person in 100 member screen to have cancer, right? So if that is the case, I tell them, like, it's so easy to develop a 99% accurate algorithm. All you have to say is no, and you get 99% accuracy. So we actually devise different metrics to actually evaluate the healthcare and so on and so forth. There are several other problems, which the challenges rather, which make AI in healthcare, particularly clinical decision support, more challenging. So, of course, when we talk about medical imaging, we are talking about these kind of images with usually radiologists are trained to interpret minute patterns which may be indicative of abnormality. The final ground truth is, of course, tissue-based analysis and so on. Because you can't keep poking to take tissue everywhere else, imaging becomes a very, very important step to diagnose any huge issue, as we all know. So again, in AI in medical imaging, from an AI scientist perspective, there's several problems. Previous speaker touched upon some of it. One is, of course, the classification, but it's not really classification. Many times it's about what type of abnormality can I pull out from here so that the doctor makes a decision what exactly is the disease that it's showing. Like, for example, this is offshoot of COVID data set, NIH data set. It had about 14 different abnormal patterns that one could extract, and you could write a multitask learner and learn all together, and then say, okay, we see there is a nodule, there seems to be an effusion. Is this, the doctor makes a decision. So those kind of problems are possible. When we actually take multiple images of the same organ, how do we align them? How do we register them? Especially when we are talking about not a visual image like this. If I'm taking a picture of this stage, for example, from different views, it's visually available that Geeta is standing here or podium is fixed, and you can actually do the registration across different views easily. But when you're looking at it from a human body perspective, which possibly changes every minute, every microsecond when the a person is breathing, etc., registration problem becomes really, really challenging and interesting from a science perspective. And localization that talked about. So exactly where is the problem likely to be? And to address one very important concern that the esteemed doctor mentioned, it's about explainability. Right? When we talk about explaining the answer, explaining the thinking of the AI model itself, one aspect I've seen based on my experience of doing the breast screening, which I'll talk about, is localization. That is, just saying somebody has cancer, you can't do much about it, right? Where is it? So that a follow-up diagnostic workup can be done at that particular point. The doctor can do an ultrasound at this, right breast, upper inner quadrant, 12 o'clock position. Then you go and do an ultrasound. The doctor does an ultrasound and confirms where it's. Something actionable should be there. Just saying yes, no, pretty much many times becomes an issue. But this is something that makes it more believable and transparent to the understanding. And several other use cases are possible. And of course, why we do it is maybe obvious. We're trying to sort of improve the productivity, making it faster, reduce misdiagnosis. Sometimes it could be calling out to, just being sort of over-diagnosis part of it, like when it's in the gray area, calling too many people, requiring biopsy is an invasive procedure. So you may have to reduce the number of false positives as well as reducing number of false negatives. Definitely is very, very important. We don't want to miss any cancer. But what actually motivates many of us, and probably the reason why I quit, was how AI-based healthcare can actually bridge the healthcare divide that exists in this world today, between developed countries and developing countries, between villages and urban areas. Even within a city like this, we have definitely two economic strata of people. And the kind of healthcare they get is hugely, hugely determined on how big the pocket is, literally. And it's not just the equipment, definitely that access to equipment and also access to specialists, given that there is a very, very important skill and takes a long time to build that expertise, and we have few in India. So this is where I firmly believe that AI-based healthcare can really help make affordable and accessible, high-quality healthcare available to everybody. So instead of speaking in sort of high level, now I go fully deep into one problem and take from the problem statement and the solution and where we are today. Breast cancer, it is the top killer in women today. 2,000 lives go every day because of this disease. It is 2,000 lives every day. Most of them could be saved. 95% survival is possible. But we end up many countries like India who has 50% survival. That means every alternate lady detected with breast cancer is dying within 2 to 5 years. In 2024, WHO data says about 2 lakh women got detected with breast cancer. About 1 lakh women died in the same year due to breast cancer in India alone. So this is the problem that we are trying to solve. And why late detection? Early detection definitely enables better treatment efficacy, reduced or improved health status, we would say. She doesn't need to sort of come back again and again. There may not be recurrence, etc. And most importantly, it also reduces the treatment cost. You know, treating a stage 1 cancer versus stage 4 cancer, the amount of money could be like 10x, and still you may not be able to save it. And this I learned from my own personal story, where I lost 2 of my cousin sisters to breast cancer, which made me quit my job as head of AI to start this problem statement, saying, can I really make something happen on the ground rather than just publishing papers, which is very nice, very important. But I said, somebody has to take that extra step. And if you look at how the breast cancer deaths, when you map it to the different continents, you will find that 71% of these cancer deaths due to breast cancer is happening in low and middle income country. Clearly is a money issue. Not only that, why are they detecting late? It is because they're not proactively doing any test. Of course, the most standard test available for breast cancer is called mammography. And if you ask this question, for any lady between 40 and 69, say, have you ever had a breast cancer screening test with mammography? In United UK, you get 71% of them saying yes, at least once in 3 years, and these kind of things. And in India, you get the pathetic 1.3%. Only 1.3% women would have ever had a mammogram. I don't want to have a show of hands here, because I think we can do that offline. But this is the statistics, even with Dr. Rajendra Badvesa, who is to be the director of TMH. It's a paper from him which says that. So the question now is, how can we enable every lady to do a screening test instead of 98.7% of women detecting their cancer by hand and then coming to the doctor when it's a 2 centimeter or 2.5 centimeter lump, which is staged as per TNM staging, they say. If it is about 2 centimeters, it falls into stage 2 and 2.5, and it depends on which other nodes have been affected. It can get to 3 and 4 very quickly, right? So lymph node metastasis we call, and so on and so forth. So we usually detect in stage 3, 4. So how can we do that? How can we reduce that? By enabling early detection screening tool. It's obvious, but the numbers are alarming. So we then double clicked on that, saying, why is this problem, right? What is so different? Of course, money is one problem. How is it? Like, what in the money aspect is bad, right? One is the machine itself is 80 lakh to 1.5 crore rupees on mammography machine, which is good, which is the one which is only proven to improve the survival rate with longitudinal study to improve by 13%, right? Which is good around the world. Now, but the machine, if it's costing 1.5 crore, how many of our small diagnostic centers can get that machine? And to operate that machine, you need a skilled resource, and she will know only how to operate this machine, and women are not coming, so there is no money, and then it increases the cost, etc. And also, of course, radiologists are the ones who will interpret it. So this is the access and affordability is the machine cost, as well as how do we do the operation. lesser skilled manner. Second is also the experience. Women who do the test once, they may not come back because they don't like the experience. And third one is the physics limitation. Mammography is basically X-ray of the breast, and when you do a mammogram, cancer would be high density, so you'll see it as white, and if the person has a dense tissue, the whole breast appears white. And so you cannot see a white on white breast. And it's not what I'm saying, it's tons of papers talking about US FDA changing their guidelines of mammography last year, saying every mammography report should have the density mentioned, and if the density is high, she cannot, she needs an additional test like a breast MRI or something, which is super expensive anyway. And you can't let women have MRI every year, even though there is no problem. So here is an example of dense tissue where even a big lesion is missed and in the X-ray of the breast, and here it is actually identified very well. Who has dense tissue? Almost all women under 45 have dense tissue, and about 50% of women above 45 have dense tissue, which means that there considerable segment of women who require an additional test despite doing regular mammogram. And this is not an India problem. It is a whole world problem. And this is a sort of a more recent analysis for us we came to know. But it means that world really needs an additional test beyond what people have been going through so far. And we said, okay, can we do something? Initially, I started off with this as a problem statement, but I said, okay, can we do something different? So if you do the same thing again and again, density-based differentiation will lead to the third problem. So we said, can we look at another imaging modality, which is more affordable, accessible? And I stumbled upon a couple of papers, which were in 1980s, trying to use infrared imaging for breast screening, right? And there were papers on it, not really very, very systematic, but at least 100, 200 people looked at it. And so they were taking a thermal imaging device or infrared. Many of you would have seen hyperspectral imager. So there they took the infrared imaging and then they were doing the analysis. And here, basically, it's one of the electromagnetic spectrum, one of the ranges in the electromagnetic spectrum, and it is light in a different format. And what happens here is based on the reflection or emission from the body, you actually, computing the environmental emissivity parameters, you can actually convert that into a temperature value, likely temperature. It's not an exact temperature, it's not a thermometry device, but it can give you relative temperatures very, very nicely. How is temperature related to cancer? So we looked at a lot of the medical science around it. Cancer cells, what is definition of cancer? Irregular multiplication of cell, which is basically higher metabolism, and that actually increases the temperature by very, very small amount, 0.02 degree, 0.04 degree. That is one aspect. And the cancer cells require a lot of food. There's more blood flow coming into them. So we said, can we model vascularity, blood flow to some areas, and can we model metabolism with these sort of high resolution thermal imaging? And of course, people had tried to use it, and manual interpretation has challenges, especially if you see here, this is a heat map. This is not like a thermal kind of any physics heat map type of thing. You will see that 34 and 35 degrees Celsius, both are looking red and light red. So it's really humanly impossible to figure out anything very, very precise in this particular fashion, unless she has a late stage cancer, which is showing up, definitely shows up as white. So we said, okay, can we use AI here and make a difference? And that's sort of the piece where what we did is thermal analytics, right? So if you start using the images, it's not going to help. So we figured out a radiometric analysis piece where we hook up the computer to the thermal scanner device itself and extract the data much before it starts showing the images. We measure or we extract 400,000, that is 4 lakh temperature points per person. And it's a high resolution temperature information with almost like 500 millikelvin, 200 millikelvin kind of thermal resolutions, using which we can actually model new biomarkers, new patterns, which world has never seen before, right? And of course, a lot of seeping through the data, going through the deep learning, but deep learning ke liye data chahiye, data was not there. So a lot of effort over the last 9 years. So with this, what we generate is a complete explainable report which says whether there's an abnormality, likely abnormality or not, and what is the probability that abnormality is likely to be malignant, and also explaining the reason for this, right? So moving forward quickly about the tech part of it, so one of the main things that happens while we use infrared imaging, is it makes the whole process extremely comfortable for the end user. Why? Because it's like a photography session. The lady sits there, click, 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 5 images are taken, and even the technician doesn't see the person not wearing clothes. It's just sort of going through, this is one of our hospitals. So she goes in, she sits in front of the device, and she just rotates to get the lateral and oblique views. 5 clicks are taken. And of course, what we're showing is a thermal image, but the temperature maps it. So I spent some time on this. What is the sort of the novelty in this whole thing? And what do I do with the 400,000 temperature points we come in? We realize just like, we have the BRCA, another genetic marker. Can we think about minute biomarkers from the thermal scans? There'll be some patterns maybe, right? Because it's not that only the cancer cell will has high temperature, because there is a convolution and convection and all of that inside the tissue. It actually spreads in a very, very different pattern. So the pattern of spreading of the temperature itself, we try to sort of model as well. And then we had to sort of bring in some kind of contrast so that this action happens sort of in a live way. So we blow cool air for 5 minutes. Just simple cool air is blown because people wear tight clothing. We don't want false positives. So with that, first thing is, in a density domain, we talk about lump, which is a hard stuff, right? In health, in thermal domain, we said there is a notional equivalent thing. It is called a hotspot. It's a common name. That is something that is high temperature. But what is high temperature? Everybody is 37 degrees Celsius. And so how do we define, right? Is it a fixed point? So earlier doctors, 1980s, when they're trying to say, okay, anything that is max -2, 32 degrees. So they were having very, very high level sort of notion of some temperature values. We said, can we learn that whether something is hot or not from the temperature distribution itself, right? So we modeled the temperature threshold learning itself as a machine learning problem, and then said, for this particular person, this threshold is hot, or rather anything above this threshold is hot. Then we defined this concept of hotspot. And there's tons of information and science available, the radiologist perspective, where they can look at a lump and say whether there's an abnormal electron. One such thing is irregularity. So we said, okay, can we model irregularity? So we mapped it to an imaging technique called fractal dimension. So it was about sort of looking at what kind of features are looked in to mammography, ultrasound, MRI. Went through the courses of these to see how radiologists interpret, getting the semantic patterns, map it to the thermal domain. And then, of course, after some time, when we started having data, we started doing a lot of deep learning networks and so on and so forth. Another important aspect is if the cancer is very, very close to the chest wall, then, it may not even traverse to the skin because we are just looking at the skin temperature. Then we started sort of staring at this kind of false negatives, deep tumors, and we figured out we can actually try to extract some high thermal activities, or rather, structured thermal activities, which ended up to be vascularity. It's not really high, but it's more like extracting thermal structural. So this again is a deep learning unit that we use, where we can actually extract structured patterns. And it turns out that this is a cancer patient where right side there is more high thermal. This is an equivalent imaging called Doppler, which actually tries to do the same thing. So all those. And then when we gave it to the end user, they actually made a lot of mistakes. So we had to do a lot of AI saying, what is the angle of incidence? Is she actually doing the 5 views correctly or not? And so on and so forth. So this is, and then once you get the network identified with deep learning, then we start modeling different parameters of it and do the asymmetry and so on and so forth. End result, about 25 different machine learning algorithms convert the 5 images into a report. Because I didn't have time to sort of go through, I just select many ML, many IM, all of those things. You see, there's all of these algorithms come together to make it happen. I know I'm running out. So in order to bring in the explainability that the doctor clearly mentioned, first thing we had to do was use the same nomenclature that mammography and ultrasound doctors are using. Of course, I have 2 wonderful doctors and mentors in our team, Dr. Ramprakash sir and Dr. Sudhakar sir, with whom we worked with very closely. And they said, okay, equivalent of a Biret scoring, we actually give a score which any doctor can understand what this the AI model is saying, right, in terms of the end result that it is predicting. And then some explainable score, is it because of the more tissue activity, because of vascular activity, or because of some milk duct issue? So we give different scores. These are all probability scores, which helps them go to the next level saying yes. And nothing like not marking it, right? Once you mark it, you can actually check whether it is right or not. So this is what the sort of whole journey has been, multiple sort of challenges, etc. And definitely we had to do clinical testing. Right, and I think one of the audience said, right, if you show an image and ask two different doctors, they may actually give two different answers because it's very, very hard to interpret an image, right? Especially, some cases will be clear, but there are other cases. So what we said is, we will compare our test with mammogram, ultrasound, and until biopsy, right? So that's what we did, and we have now 58 papers. I'll just flash a few results in the interest of time, and happy to share with you. So we focused on sensitivity. That is, how can we give a test in a way that there are close to zero false negatives. That is, when we say no, it's a no, highest negative predictive value, and so on. So we compared with mammography, ultrasound. Then we also had to sort of look at, if mammo is done, if you add this, will it add value or not? So different kind of settings had to be done. So we made this as, if thermalytics is saying yes, then second radiologist will come and see the mammography, so that we can go ahead and pick up even in dense tissue. And then we did some performance analysis in pure dense breast, young women, as a meta-analysis. That was also extremely promising, and published. All these are peer-reviewed journals that is published in. And just to give one example, this is the dense tissue where mammogram is not able to make out the difference between right and left, and then physiologically the thermal scan is able to determine something that was not, is not visible, and it's just a limitation on the physics part of it. We are now, of course, done several rural screenings as well. We have a Nature Digital Medicine paper, which talks about how 15,000 women were screened, and only 3% of them had to be actually brought back to the hospital to do a further detailed analysis, where, of course, we brought many more. Not everybody was going to have cancer, but we are sort of working on them. So this is one of our flagship projects. Where we actually determined 3 times more cancer detected with this. So I'll stop here. Would love to answer any questions you may have and thoughts on this.
Thank you very much for that. Mic's not working. Thank you very much for the lovely talk. Unfortunately, we run out of time for this talk. So
Okay.
The questions, so they started at 25, I think. So we're going to have the questions over in the coffee session. So let's thank Dr. Geeta once again. And invite.
Okay. Sorry, I thought it was 20 minutes. Okay.
They changed the setting at least.
Oh, okay. Okay. Thank you. No worries.
Thank you very much, sir.
Yeah, sure. No problem.
Sure. Thank you. Okay, that's fine, yeah. So let me begin by thanking Professor Somdatta and Professor Narendra for this kind invitation. It's a pleasure to be here. It's okay. So I thought I will talk a little bit about how we are using AI, especially the generative AI technologies, for asking some basic science questions. We do have a... Oh, 20. Thank you. Thanks, that helps. Yeah. So, of course, AI has been extremely useful in the end stage application, as you heard from brilliant talks from the previous 2 speakers. We do have in the lab precision medicine-based applications, but I thought today I will tell you one story about how we can use a basic
question using AI. So much of the work that I'm going to talk about is done by my graduate student Arnav from the bioengineering stream. So I'm going to tell you about how we decode immune response patterns, the complexity that are there across diseases, using, of course, physics-based information, large data, especially transcriptomics, and AI models, right? So let me begin by just reminding all of us, I mean, this is not new to anyone, I'm sure. So the fact that humans are afflicted with a variety of diseases. And when you think of disease, the first thing you think of is the organ, perhaps, the symptoms, which doctor to go to, because it's so very different. And then, with age, perhaps the cost and practical aspects like that. Let me give you a different perspective. Of course, the whole lot of diseases, the point was not to give you a lot of information on this. It's just to remind ourselves about the space that we are working with, really. So thousands of diseases, out of that maybe hundreds are well characterized, and some of them are very frequent. Some of them don't even have a name, and you just assign some name to it, depending on various other things and so on. I want to give you a different perspective, a molecular systems perspective of disease. Something like, perhaps more appealing to engineers, I would think, than conventional biologists, but it is nevertheless very important to think about it like this. And now we have the means to think about it. It was not possible. Let's say when I began my career, people would have laughed at me if I had started with a question like this, because it was just not practical to do that. But today we can actually do this. Right? You think of it as a box, not a black box, as a transparent box, perhaps, where something senses the pathogenic triggers, right? Whether it's a mutation that's happening in cancer cells, or it's a pathogen like a virus or a bacterium or whatever that may be, and molecules from that. So that's a trigger. And then something senses that and detects the trigger and relays that information. All of these are to various protein molecules that are there in our human cells. So to responder machinery out there who then respond. I mean, this is oversimplification of the whole process, but that's what it is. And what happens at this molecular level must be felt at the cellular level, at the tissue or organ level, which is what actually gives rise to symptoms at the organismal level. So the clinical manifestation that you see is a result of all of these things that are going on. Whether it's in a model system like a mouse or a human, it's the same story. So I would like to think of disease as a function of molecular aberrations at the tissue or organ or whatever, and physiological perturbations that goes on, which are symptoms. But the story is very complex because there is a pathogenic trigger. It's not a linear pathway that goes into aberrations and symptoms. Because even when there is disease, people are still alive, cells are still functioning, metabolism is happening, you're thinking, eating, drinking, whatever, all that is happening. So there's compensation that is going on. The local perturbation, the global perturbations. There's a compensation, homeostatic correction, repair, and so on, and metabolic rewiring, reprogramming, and so on. And the outcome is a combination of whether there's perturbation, compensation kind of a thing, and recovery, and whether it goes into a chronic state that we live with. I don't know how many chronic illnesses each of us have. We really don't know. That doesn't manifest actually as clinical symptoms. And whether it progresses into something active. So why I'm giving you this picture is just to drive home the message of complexity that's out there. And let's just go back one step. We have like 37 trillion cells, something in that order in the human body. And each cell has 20,000 genes. Even if you take only coding genes, orders of magnitude higher number of transcripts of each of these genes, and the number of protein molecules, a large number, many zeros out there. And if you look at interactions among them, it's orders of magnitude higher than that. So it's a very, very complex picture. And this is changing dynamically. We have seen changes within a few minutes after a trigger and so on. So those kind of changes happen. A few minutes or even earlier, maybe. Why the complexity, the homeostasis includes all of these things: feedback, regulation, cooperativity. I mean, I don't mean this as a classroom lecture, but all of these things necessarily are going on out there. And the result of this is emergent behavior, robustness, coherence, adaptability, and so on. Okay. So again, the point I'm trying to make is it is not sufficient to look at single molecules or single pathways at a time. We really need that helicopter image and be able to dive deeper as necessary. So systems biology is really essential. And the way to do, one of the ways by which you can do systems biology is collect large data, which fortunately is now possible with all the omics and related technologies, make mathematical models, simulate it, and then do that. So this was what we have been doing in the last 20 years, in fact. In fact, it's, people talk about fourth paradigm or fifth paradigm. It doesn't matter what we call it. So we want to harness the power of big data, bioinformatics, systems modeling, and AI, all in sync. Okay. So at the end of the day, I'd like you to just remember that they're complex patterns, and recognizing which pattern is associated with what, or identifying these patterns, is one of the goals in this field. So with this, equipped with this kind of understanding, now we thought we will ask very specific questions about diseases. The same human system responds to a variety of pathological triggers. We are able to come up with a response to different types of diseases and then solve it to various levels, right? The same set of 15,000 to 20,000 genes present in the cells respond to these triggers. And each person who sees this trigger responds to it in a very similar way, which is why we can recognize the disease as a disease. Of course, there's heterogeneity within that, and there are subgroups, and that's where precision medicine comes in. But nevertheless, you can recognize diabetes as diabetes and cancer as cancer. So what gives rise to such precise molecular perturbations in the cell? And how do our cells sense these pathological triggers and make sure that the downstream processes are so precise? How do they transmit? What is the mechanistic understanding of this? How is a phenotype produced very precisely? That's what we're trying to show in the image out there. Now we have a problem. We want to study many diseases. Each one is a different organ, different tissue. We can't compare apples with oranges, even if they are human cells. You just cannot compare liver tissue with muscle tissue and then come up with an answer that is scientific. So to compare all diseases, fortunately, we have blood as a tissue. Blood carries disease signatures, which you can read out from the transcriptomic data. So that was what, and there are many publications, many papers, a lot of experience in this thing. So if you see the image on the right-hand side out here, each column out there is a transcriptomic data of one patient. So the colors actually represent out here whether it's overexpressed, underexpressed, or no change as compared to healthy people. Some of these are healthy individuals. Each column is a patient. Some of these are, I've taken TB as an example because we work a lot with that. Tuberculosis patients out here, blood samples from tuberculosis patients. And these are treated at various time points without telling the algorithm anything about the condition of the person. If you agnostically cluster, you'll see all healthy individuals going into one side and all the TB another side, and the treated ones, depending on the success of the treatment, group somewhere closer to one of the two sides, which means that the transcriptomics contains enough information to indicate the disease status. And this is not just in TB, it happens in cancer, it happens in autoimmune diseases, it happens in metabolic diseases, and so on and so forth. So blood is an accessible and unifying tissue that reflects disease states. Blood transcriptomics successfully classify healthy from disease, and transcriptomes are beautiful. I like them a lot. The measurements of entire genomes. So it enables biology at scale. Now we have many advanced technologies, spatial transcriptomics, single cell transcriptomics, and so on, but what I'm showing you is just the bulk. Now let me quickly go into the diseases that we've been looking at. So we obtain blood transcriptomics that are publicly available because we have small data sets here and there that we got from clinical studies, but that's nothing in size as compared to what you have out there in the databases. So that's what we take. So about 53 different diseases out here, many data sets, total number of samples in thousands, including controls, pathology matched controls, healthy controls, and so on. And these data set represent several major disease types. So we thought it was a good starting point to do that. The first image that you see is based on what's called disease ontology. It's another database where people look at the organ, the etiology of that, and so on, and then come up with anatomy-based hierarchical disease classification. So we asked a simple question: if we take transcriptomes, will we be able to reproduce this classification that's well accepted in the medical world? Okay. And indeed, it works out quite well. Of course, we have different measures for comparison and so on, which I will skip in the interest of time. So that was the starting point. Now, we wanted to leverage, to ask these questions, which are so complex, we wanted to leverage the system. I mean, we were able to do that because we had systems modeling methods in the laboratory at different levels in the biological hierarchy. I won't dwell on them, but for each purpose, for each sub-question, we have a modeling type that is available. Many of them are available publicly, and a large number of people who have worked on these in the world. So the method that we used quite heavily here is to use a knowledge graph of protein-protein interactions in the cell, which includes physics-based interactions, includes bioinformatics predicted over the physics information. So it's somewhat like a network, a large network, a roadmap of a large city like Bangalore, a messy city, I would say, in terms of traffic. Something like that is what you see. And to mine out very specific interactions that are relevant, is a challenge. So we came up with graph theoretical algorithms, which will tell you what are the changes in this big city, let us say between 8 a.m. and then 6 a.m. in the same city. Where is the traffic going? Where are the traffic perturbations that are out there? So our algorithms capture some of those and then get insightful ideas about which pathways and so on and so forth. So what we had as a number of publications from the lab on this. So what we had as a starting point was RNA-seq data for each of the diseases, contextualized graphs, genome-wide knowledge graphs, and this is the perturbation networks that came out of the algorithm. And even this was so complex that we could not make sense out of it by manual inspection and so on. So we also have functional enrichment and so on. So what we found from this was across all diseases, immune processes ranked as top perturbed. And out of that, it was the innate immune pathways that were top perturbed. So if you see these things out here, they're present in more than 90% of the diseases, even if they are not infections. Right? So that is what you actually see. So a human innate immune system is well understood. There are a number of pathways. It does a lot of functions. But one thing that I want to draw your attention to is the textbook image says it's non-specific protection that detects danger and so on. So is it really non-specific is a question that we asked. And then we wanted to understand if innate immune system is at the common core of all of these diseases, can we identify if there are any central players in them, right? Who is allocating basically a response pathway to this particular pathway or that different one and so on and so forth. Are they central players is a question we asked. To address that, we came up with this AI method, a graph neural networks. In brief, a graph here means the protein-protein interactions for us. And the weights of that initially informed by transcriptomics data, but we want to learn based on attention, just like, I think attention is a well understood problem now. Can you go on increasing the weights if they are important for the problem to solve? So that's what we are actually looking at. So it's a big improvement over traditional GNNs with the attention-based ones. And we thought we will do it just like humans learn, do it at different levels, go to primary school and then to secondary and then graduation and so on. And that's what exactly is happening out here. There are different levels of this is going on. Okay. So Arnab actually devised a GNN, a graph neural network at different layers, 3 different layers out here. I will keep the technical part short. And at the end of the day, we were able to identify a panel, a small panel of 23 genes as central tuning factors. Now, these tuning factors are not direct sensors like pattern recognition receptors and so on, but they are central. If you change their expression levels, you get a different tune pattern. But it's a combination of them. It's not a single one at a time. Not all 23 are present in everything. A subset will be present in any given disease. Now we asked a question, if we just take this 23, can we reproduce the classification pattern of diseases? And that's exactly what you see here. We call this T23 set. These 23 are central. and they are the most influential, which is what was identified by the AI algorithms. They reproduce largely the same clustering pattern that you had with the disease ontologies or with the whole transcriptomes, indicating that, of course, there will be some differences. It's not identical. It may even be better in some ways, but it is sufficient to get this broad classification going. So that was really interesting for us. Now you ask, what are these 23? How do you actually make sense out of it, and so on? So we looked at a topology-based node annotation. Where are they present in this network? Are they in the central core? Are they in the next neighborhood, primary interactors, or are they satellites somewhere along the way? First satellite, second satellite, and so on. So those are the colors that you see out here. Examples of them I've taken again tuberculosis and cystic fibrosis, which are very different diseases really, but there are some central players in red. These are gene names, so that are common to both of them. The same genes are present, but their neighborhoods are different, which is why they're such different diseases out there. In fact, the tuned patterns provide insights into the differential pathogenesis mechanism out here. It's just a little snapshot with the different genes and so on. It may not make any big sense to you from far, but they're very insightful because each one has a gene name and interaction, and which is these red ones you see out here are the nodes which are part of this 23 gene panel and so on. So in fact, TB shows a system where compensation can genuinely win because it can push it into latency, whereas cystic fibrosis shows a system where the initiating lesion structurally prevents compensation, because even the neutrophils, antibodies, the adaptive immune responses can become pathogenic. So we're able to dissect pathogenic from protective consequences that are going on in this system. So that can form a separate question, which we plan to take it up in the future. But you get like a very, very insightful, detailed resolution of information out here to say what is the trigger, who are the responders and what is the response, who are they relaying it to, and what is the action that you see at the tissue level and at the organ level. So in fact, let me stop with a summary here. So what our study has of learning from pan-disease transcriptome, what it shows is that innate immunity has specificity. It's a common thread across major diseases, and it's well supported, of course, by converging evidence. You'll have to synthesize the evidence from individual studies because nobody's asked this question. It is, like I said, textbook information that innate immune responses are boring. They're important, but boring. There's nothing exciting about them. But that's not really the case. In fact, in a separate instance, we have T-cell mediated, CD8 mediated immunity-based work in the lab for cancers. And we are seeing differences in survival when innate immune response triggers CD8 responses in a certain way, or whether they push them to exhaustion or whether they push them to being active and so on. And that has a survival implication out there. So for this, again, of course, innate immunity and inflammation go hand in hand. And if you can think of innate immunity as the sensors, sense and respond, inflammation is like one of its most visible outputs. Okay. Now, there are GWAS, genome-wide association studies, where you associate genomic changes to transcriptomic changes available for many diseases. And again, we find that immune inflammatory pathways are enriched in almost all diseases, which ties up with the data that we are looking at. So our study does indicate specificity in innate immune patterns, not individual genes, but as a pattern, as a subnetwork, with each disease exhibiting a characteristic pattern. And we find 23 to be at the core, and they can be called as tuning factors, which gives rise to specific tune patterns. And then as a last comment, I want to say that AI technologies have high potential in driving new basic science discoveries and finding generalizations in biology, perhaps as much as applications, downstream applications. Thank you for your attention.
You mentioned modeling knowledge graph for the physical interaction between the protein molecules. So I'm assuming that is a very large data. So instead of that, if we forego the pathways and instead model it as a correlation graph, between the first few molecules and last few molecules, or gene expression as such, would that provide a similar kind of accuracy or result?
Got it. It depends on what you mean by correlation. Correlation is very context specific, right? So our idea was to start unbiased, don't have any favorites, don't assume one hypothesis and chase it, but instead derive hypothesis and later verify that. So that was our idea. So you can do that, but you will require some data for those things. Yes.
So, very nice talk, Suma. You just have one quick question, which is that you discovered these 23. They are all part of the innate immune system. I'm thinking about application, and you gave an example of cystic fibrosis.
Right.
Why would I apply the 23 transcriptomic signature as opposed to Delta F508? That's my question. So when would I use your system?
Right.
You're learning, of course.
Yeah.
So if you have an unknown disease, maybe you can.
Yeah.
But the examples that you gave, we know what the players are. So
Right.
why would a clinician
No.
want to do 23?
This was only as comparison to ground truth, right? So you have many
What I would
new
have liked to see
findings.
is something that's not known in medicine.
Absolutely.
And you have discovered it using your 23.
Absolutely.
That would be a wonderful.
Absolutely. First of all, you identify the entire process, like the sensors, which combination of genes are basically involved in sensing the pathogenic trigger. And we are also, working on quantifying the response of sensing and transmitting and relaying and so on. So these, at this point of time, I don't want to claim that, this is applicable directly. This is more fundamental understanding. But what will come out of it, I know because we are working from the other end as well, is applications for subtyping a disease. So looking at what is protective versus what is pathogenic as a consequence. So what is a compensatory mechanism? So, and treatment can be tailored based on that understanding. Yeah.
Thanks for a very interesting talk. How do we validate this? Do you have a plan?
Yes.
How do we do that?
So it's not a simple validation like we would do for, say, a single gene biomarker, because this is a systems profile. The validation that I showed you to some extent was how genome-wide association studies have found mutations. Can we synthesize the evidence that is coming from multiple types of data and then put them together? Is there coherence, is what we are trying to ask. And then, of course, there can be more focused experimental studies to validate these things. But when I sort of come up with an idea of how to do it. Yes.
Good morning, ma'am. I have a question. Like you mentioned, like every disease has its tune pattern, right? So if let's say somebody has two diseases at the same time, so how does the AI model help in differentiating between the two diseases, and how does it like not mix them up?
Right. So so that becomes a new type, a new phenotype, which is a combination of disease A and disease B. So again, data is important for any machine learning model to happen. If there is data of, let's say, hypertension and diabetes are so common together, so one has to train with a new label out there, right? And then there are deconvolution methods to do that. It all depends on how much training data you have.
Let's thank Dr. Suma again for a wonderful talk.
Thank you.
Thank you very much. Dr. Oishi Mazumdar.
Good morning, all. I'd like to first thanks for the opportunity. I'm really honored and humbled to be here and present our work in front of such an esteemed audience. This talk, we all know how AI has transformed the healthcare space, or is transforming the healthcare space in the past decade. Now, in my talk, I'd like to touch upon a new concept, which is the concept of digital twin, which may be the next chapter in the way AI is transforming healthcare. And this is not about only like AI has been really good in identifying patterns from a large set of data, and it has finds its usability in screening, early diagnosis, risk stratification. But now moving forward, we are looking at a really precision and personalized healthcare, a future where healthcare will be personalized and not one size fits all. And the concept of digital twin actually fits over here. So in the present domain, we know that healthcare is having a shift from the conventional traditional care paradigm, which is hospital-centric, event-centric, into a more emerging care paradigm where care will be beyond worlds, like you will be having the care essentially not only at hospitals, but even at your home. So there will be continuous monitoring. Not only that, the care will be patient-specific and it will be personalized and precision. Now, in this transformation, AI has played a very important role, which is mostly in screening and detection across modalities, enabling pattern recognitions, and advancing towards interpretable and explainable models. But still, there is a gap when we are envisioning a future which is going forward to the personalized medicine and care beyond worlds. The gap is that although we understand the disease and we can predict that which percentage or which strata of person is going to have this disease maybe at an earlier time, we actually cannot define the underlying physiology which is governing this disease. And the models and response to intervention. So just to enhance this point, just take a very basic example that an AI model has predicted ECG-based risk prediction. And using explainable models, it has also predicted that why it is saying that this patient has got a high risk. Maybe it is pointing towards the QRS direction or certain ST segment elevation, which you could link to myocardial infarctions or something, heart rate variability, etc. This is important. It is identifying patterns in signals, giving you cues that why it is telling you that, and driving certain sections of patients for early detections and early diagnosis. Now, what clinicians do? They already have this disease map in their mind. They see the signals, they see the report, and they already know which patient is at high risk, what therapy is to be given, when the patient needs monitoring, etc. Then what is the problem? The problem is that the physiological state is not completely explained. And as we have heard from experienced doctors themselves, that experience actually varies. A very well experienced clinician will have his understanding at some level, whereas the juniors will not have that same level of understanding. Now, how can we bridge that experience driven and variability into the model? So the limited ability to track, compare, or simulate is another thing that we're going after. So we know that the patient has this disease, and some doctors or some caregivers will have a standard approach to therapy. Now, what if certain else happens? How this two diseases is going to interpret? Or what if I want to simulate and see 10 years down the line or 5 years down the line what is going to happen? Or if the subject or if the patient is doing certain strenuous activities, if something else happens, how this intervention may change. So from explainable and predictions and implicit inference, we want to go towards explicit quantitative representation of physiology, and this is where biodigital twin comes. So what actually a digital twin is? So digital twin has been, or it's a newer concept in healthcare, but it has a sufficient footprints in other engineering domains. So a very well-known engineering domain where it has application is in aerospace, where jet engines are regularly monitored and they go for their evaluation patterns through digital twin of the engines. So basically it's a replica, it's a virtual system representation of the physical system that evolves with data and enables continuous monitoring, prediction, and simulation. So a very important aspect of this digital twin is that you can do what-if analysis. If you have a virtual replica of a system, that can be any system, any engineering system or healthcare system, and you can specifically do or ask what-if analysis. What if this stress level is increased? What if the drug dosage is changed? What if there is another incident that is happening? How the physiology is going to evolve at that, and how do you take decisions at that? So, so digital twin, basically, it represents the system, not just observations. We get the observations from multiple amount of sensors. Now, how to bind all this sensor data and to have a representation of the physiological system itself is the question. So, moving into the healthcare space. Now, how this digital twin is actually transforming this healthcare space. So, in healthcare, unlike any other engineering domain, the data modalities are sparse and incomplete. You actually don't have sensors attached to human body at every time. So these are all mostly sparse and multimodal data. So what we have is that from subject or patients, you have streams of data. It can be imaging data, it can be time series data, it can be blood reports data, many sorts of data that comes in the respective modalities in sparse way. Then we have an AI analytics layer which tries to make certain decisions or certain pattern which tries to take from this observable type of data. And there are processes which make risk prediction, biomarker extractions, etc. Now, what the digital twin layer does is that it integrates all these things into a physiological model layer where embedding of this domain knowledge and system mechanics happen with the mechanistic model of how the physiology is going to behave when we observe such data. And once you have that model in between, many different types of applications like personalized care, simulation of what-if analysis, predictions, and actionable insights is possible. So in a nutshell, digital twins integrates data, AI, and physiology to create a living patient-specific representation that can be observed, updated, simulated, and can be guided for better decision. So essentially, from analyzing data about patient, it's going to represent the patient as a dynamic physiological system. And there lies the difference. Now, when we look at healthcare system, we all know that it's inherently a multi-scale system. So it actually starts from the genomics and metagenomics layer, like ma'am clearly explained. So the disease actually starts at the genomics level. And for the understanding of the disease, we need to look at the genomics level. From genomics, there is the cellular pathway, then from cells to tissue. But the actual manifestation of the disease or progression of the disease or the action actually happens at the organ level. So the basic entry of the digital twin point is basically the organ system modeling. Just by doing a cell level analysis or genomic level analysis will not give you the understanding of how the disease is progressing or how the organs are actually functioning or responding to that disease. Hence, the entry point of most of the digital models are via the organ digital twins. And this is where TCS is also working a lot. We are actually building a lot of this organ digital model, cardiac twin, brain twins, pulmonary twins, gut twins, and musculoskeletal twins, to name a few, where we actually study the effect of disease progression or how the functioning of such organs are happening so that we can link the observation to the disease to predictions and simulations of different what-if analysis. So organ level digital twin gives us the entry point to represent this multi-scale physiology with long-term integrations towards whole body system. Now, just to give you a broader example of what this organ level digital twins are doing, I'll take the example of cardiac digital twin, which we work upon, and give you a few examples of where this cardiac digital twin have seen applications both in healthcare and in wellness. So what this cardiac digital twin is, it's basically a computational model, a biophysical computational model, which models the cardiac functionality. Now, when we say cardiac functionality, we all know that its functionality itself is multidimensional. Like you can explain the functionality from initiation of the contraction of the pulses, how the ECG actually generates, to how the excitation actually travels through the tissue and brings about the complete contraction and relaxation of the heart. The contraction of the heart itself then pushes the blood through our circulatory system, which is the complete function of the heart. So basically, there is an electrophysiology functioning which generates your transmembrane potential or the initiation of excitation. the excitation travels through the cardiac tissue, which brings upon the contraction. So there is an excitation-contraction coupling which happens through biomechanics, which then brings upon the circulation of blood, which is the hemodynamics functionality, like how blood passes through the different phases of your cardiac cycle, how it moves from atrium to ventricle, how it then moves from aorta to the full circulatory system, that we model through the hemodynamic functionality. And closely linked with this cardiac functionality is also the airflow dynamics, because the cardiac functionality, when we go to model cardiac functionality, there is the systemic circulation as well as there is a pulmonary circulation which needs to be connected to have the complete circulation overview. So the cardiac digital twin, basically it encompasses a 3D structure which we generate from CT or MRI images, and we have built this functionality through different types of computational models. Now this model, computational model, once it's ready, it can provide or simulate the effect of different types of physical, pathological and pharmacological interventions, providing mechanistic insights into the cardiac disease progression, which gives the pathway towards personalized and predictive therapy. Now, when we say disease progression, basically we have modeled the progression of various types of disease through this type of physiological modeling, like valvular disease, different types of arrhythmia, myocardial ischemia, how the genesis of this disease happens, and through the myocardial functionality, how it expresses and how actually it affects a person, that journey has been traced. So once you have this type of functional model in place, different types of application can be thought about. One is this disease progression and analysis, where medical persons can see that if the disease is progressing from moderate to severe, then what are the changes it's actually happening in the parameters that they know to be important. So we have different types of parameters from pressure volume loops, like ejection fraction, cardiac output, which can be generated from such models, which gives them an understanding of what actually is changing inside the physiological model. These models are also used for therapy optimization, device tuning, cardiac rehabilitation, personalized therapy management, and also has been thought for virtual trial and drug screening using cardiotoxicity as the measure. So there are lots of applications, but today I'll touch upon two specific. One is the use of such digital twin platform or cardiac digital twin in simulating or pre-planning surgery for congenital heart disease. And the next one is a wellness application that will come after. So this particular application, if you see, surgery planning for congenital heart disease. So congenital heart disease, mostly the affected persons are pediatric population, very small children, where the heart size is very small. And they have certain structural abnormality for which they need to undergo surgery, and the surgery actually happens within the first 2 years of their life. The challenge is that because the structure is abnormal, which again gives the hemodynamics actually becomes abnormal because the structure is abnormal. So each and every heart is very unique. So for doctors, many times it becomes very difficult to perceive that what the change is, what change is happening and how do they counteract the change. So there are surprises during the OT, and the problem here is that the child is actually in a bypass during the surgery, and the surgery time matters. Now, if there is not sufficient pre-planning, what happens is that the surgery time extends due to this occasional surprises. The utility of this digital twin now comes in is that you have the structural and functional model of the child that you want to operate, and you know what is the difficulties over there. So from MRI or CT, you will get the structural, and from computational fluid dynamics elements, you will get the fluid structure interaction. Now, not only the surgeon knows what the problem is, but they can have endless amount of simulation of what will be the best technique to solve this problem. Not only that, say the patient heart will grow in that age. As the patient will recover and grow, the heart muscles and heart tissues will also grow. So the surgeon also needs to know that the patch they're putting, will that substantiate when the cardiac flow or the volume will actually increase as the patient goes up. So there can be endless amount of simulation with which the surgeons can do and predict that which is the best way forward, which not only enhances planning, but also supports the idea that the congenital heart disease can be tackled in a much better way. So from static interpretation and fragmented insights that the doctors have today, this type of solution can give them a dynamic simulation for confident decision. Now moving from a clinical case, how this cardiac digital twin can be used for performance and recovery analysis of athletes. So this is a wellness application that we tried on New York Marathon 2024. Several participants actually gave us the data to create a digital twin of their heart. And from their data, from the digital twin of the heart, we actually predicted different latent space. Like you have enormous number of sensors today that the athletes can wear. You have digital fitness app, which gives them the heart rate, the speed, the MET levels that they're crossing, okay? But what the sensors or the observations will not tell you is that how the cardiac energetics or how the your cardiac behavior is changing or responding to the load that you are like facing when you are running. So for trained athletes, there is a term which is known as athlete's heart. So athlete's heart is basically a remodeled heart because of the trainings that they undergo and the large volume of trainings and runnings that they undergo. Their heart chambers are usually enlarged. So that is a benign condition, but many times it actually camouflages some serious condition inside. So nowadays, the marathon runners or the elite runners or the elite persons in sports, they need to have this cardiopulmonary assessment time to time to check whether their cardiorespiratory system is functioning well when stress is applied. So what this cardiac digital twin is giving them is that it's giving them a tool in real time, and they can upload their running data and they can see whether how their cardiac energetics was, how their cardiac system is like responding to load, and most importantly, how they're recovering. How is the recovery pattern, which is actually not visible only from the observations. So from the cardiac digital twin of the runners, you are getting latent space variables like cardiac workload, energetics, hemodynamics response, the recovery rate, how the heart is actually coming back to this normalcy. And this allows for actionable outputs. Like from these values, you can generate, or coach or an expert can generate training load adaptation, which runner, how much load, how much weekly load they need to train on. If their recovery pattern is not that great, what can be suggested to make their recovery better so that their performance gets enhanced. So from observation of the signals from sparse sensors, we are actually moving to estimated physiological state to personalized action, which the athletes can take to enhance their performance. So when I spoke about the last example, the digital twin in action was the cardiac digital twin. But we all know that the response that we observed was not only meant or not only due to the cardiac system. Basically, when a person is running or when there is an exercise or stress, different types of organ systems comes into play. Just for in this example, the cardiac twin has a very interway or a connected pathway with the respiratory twin, which actually determines the amount of oxygen intake, which in turns again is related with the musculoskeletal system, which is driving your demand. So there needs to be a closed interplay with all this system. And if you see about how the cardiac twin is maintaining your blood pressure and balance, it has to be again linked with the autonomous regulation system or the kidney system. Yeah, I'll just wrap up quickly. So what now is present is organ digital twin at individual level, but where we need to go is that simulation of different organs in together so that we get not individualized system, but a systematic representation of the human body itself. And once we get to that system integration view, we can have a whole lot of application, which can be whole body response to therapy like drug intervention effect, recovery and resilience modeling, cross-domain decision making, etc., which will become possible once all these individual organ models gets interconnected. the main challenge that we face in making this digital twin is that one is this sparse amount of data, but the next one basically is the inverse problem. So physiology inherently, it's basically an inverse problem, and all the observation can link into one or similar type of physiology, which is not the case. So the solution is never unique, and you need to have certain a priori conditions and constraints to be put in your modeling platform so that it becomes the inverse problem could be solved in some way. Also, there is a problem of validation, like how to validate, in which context to validate. The model will always churn out data, but how do you actually link back and say that this physiology is correctly represented? So in these directions, there has been to outcome the challenges. One particular direction is that integration with foundation models. So AI, particularly in recent years, very phenomenal work on foundation models is going on. So from the foundation multimodal data, if you can learn the latent space and include that in our mechanistic physiological model, it's hypothesized to give better results. So this is a research direction that the consensus is going on. And for certain global initiative, if you're interested, you can see the Physiome project, which actually speaks about that. So there is a virtual physiological human project going on across European Union countries who are trying to build these organ level models into a full physiology model, just to enable applications like virtual testing trials and what if simulations. So we understand how data are modeled from the real world scenario, and the AI has been really a translators and tool to understand that. But what we actually need now is a way to move from this understanding to guide personalized and predictive care. And so the future of physiology-driven healthcare will be enhanced through these biodigital twins, so that we can truly have continuously understandable and individualized physiology-guided platform, which will enable precise and personalized care in future. So with that, I conclude my talk. Thank you.
Thank you, Oishi. A couple of quick questions.
The digital twin is constructed based on multiple people's data, or is it one person's data?
Digital twin parameter optimizations are based on multiple people's data, but when you come to personalization decision taking, one people.
So if it's multiple people's data, the input data matrix will have lots of holes depending on from whom you're collecting the data. If it's a patient, you would have MRI data. If it's a normal individual, you would only have wearables data. The MRI data would be missing most likely. So how do you take all of that into modeling?
Yeah. So for disease-specific cases, or for the athlete case that we talk about, it's single person's data driving that model. So you have digital twin of that particular athlete driven by the ECG of that particular patient. The cardiac structure is from the CT MRI of that particular runner, and the personalization from echo of that particular runner. So there is no intermixing of data in that sense.
So every time you have to construct the model.
So there is a global model which takes data like the digital twin heart of the heart. The hemodynamics model is a global model. You have to tune or personalize each time when you take new data. Yeah. So that personalization is the tuning process.
So who owns the digital twin? I mean, this is an ethical minefield. I mean, given insurance and various other things. So who actually has ownership and right to use the digital twin?
Okay, right now we are not in a space to sell the digital twin as an entity. So that question, maybe I don't have the answer right now, but the onus will be on the owners itself. Like if the company is providing that platform or that tool, then it will be on the company itself to defend. Yeah.
Thank you very much. I'm sure there are lots of other questions, but we'll pause in the interest of time. Let's thank Oishi again. And now I invite Somnata to give closing remarks and conclude the session.
I suppose I didn't want to miss the chance of coming on the stage. You know, in a when you organize a session like this, obviously 4 talks don't represent a particular area which is so new, which is novel, and which is fast moving and growing very, very well. Also, it's an interdisciplinary area, so you need people from absolutely from omics and experimental biologists all the way to people who are clinicians and probably have never, except when they studied, have looked at DNA, RNA since they have been doing their medical practice. So it is not possible to have everybody or all kinds of people representing their case. So I think one of the most important thing that we want to project is it is very necessary that we continue with these kinds of programs or these kinds of in workshops and schools and things where you need data scientists having clinical background or clinical training and clinicians having basic training in data science, which is almost impossible to think of today, except a few institutions. When if you are a zoologist, you're a zoologist. And if you are a mathematician, you're a mathematician. Okay, so what I would like to do, and I must tell you that what I'm reading out is made by AI. It's not written by me. And I really loved it, so I'm not going to say much beyond this. So instead of summarizing what you heard just now, which all of you, I'm sure, have in mind and would be discussing over tea, I would like to spend a few moments on what we did not get to. Not what we heard, but what we did not get to. One of the things we did not get to, which is extremely important, is data. And we did have a talk on the data by people who are involved in the government initiative of setting up Indian data. But as I call it, it's karma that he could not come. But it is the trust and the validation of data is the scaffold on which the entire area rests. All the things that you heard, it starts with validating their model, validating their twins, validating whatever one is talking about with existing data. So do you trust the data? You already heard that depending on what is your training, you may or may not even be able to distinguish between two X-rays or two MRIs. So having trust and be able to validate the data is something that needs a separate, altogether meeting probably, which would talk about such things. Now, clinical validation at scale, that is if you are trying to see whether this particular thing is going to work on a population, you really need to have prospective trials. And that is where the regulatory pathways become very important, in the sense that how do India's, for example, in the case of India, how do India's regulators, hospitals, and insurance people, how do they evaluate and approve these tools? That's something very important. And that's where, of course, the government intervention becomes very important. Another point that has not been covered is equity and health economics. Health economics is an important area these days. Will these tools remain at the level where people can only pay, or will it reach the levels at the rural areas where at low resource settings, who bears the cost of building and maintaining them? Who's going to take care of that? That's an area completely different, but health economics is an area where we need to worry about. Data governance, explainability and clinicians' trust. Do the clinicians trust what is being built? And it's important to be able to trust what is finally coming out of a machine. And that's where experience matters a lot. A doctor, a clinician who has long experience and somebody who is not makes a difference, because that's where they have to make a decision at high stakes, at the level where there is high stakes. Data governance beyond consent. It's one thing to take the consent of me and use me as an experimental tool. But it's another thing to be using interoperability of data and security and long-term stewardship of the data, health data at a national scale. And we have regulators in India who hold to their heart all the data, medical data they have, which sometimes they don't even share with others. So a lot of us who are outside that regulatory body don't even get to validate our models because we don't have access to data. So it's a very important thing for people like us who are involved in modeling to be able to use data which is already there in Indian population, but we don't have access to it. There's, from the computer science angle, generative AI and large language models, these are very critical. There are several models that were compared in one of the talks. And it's a very fast-moving area. And we did not really talk only about those models, but one can do that, and probably there'll be future meetings where they will discuss about that. One very important thing which I personally am involved or interested in is workforce and training. You need to have a clinician data scientist hybrid talent in this field, and we need to train such people. We need to train our students who actually are interested in these things, but then they are pushed into specific specialization where they then forget to even think about all these things. So we do need meetings like this, or workshops, or schools like this are going to be very helpful in this. How do we take this forward? Obviously, part of that has already been talked about. One thing that I think is important is, instead of each one of us collecting a small set of data and keeping it with us, we should have shared infrastructure. In the sense, the data collection has to be much more widely available to everybody, and people, there should be a common auditable benchmark of diverse data. You see, when one talked about multimodal data, if you're in the field, you know what it is. It goes all the way from a DNA sequence to an X-ray plate. So there are very few people who understand all these different sets. So all these multimodal data and benchmarking them is very important to have a common approach towards it, so that the pipeline is available and validated. I think the companies do that, but pair clinicians with the AI groups from day one of the project. There's no point in having the clinician, give the clinician an algorithm and say, okay, you run this and it will tell you what it is going to be. You need to have their input, because that's and you need to have their input right from day one. So that collaboration is extremely important. Invest in Indian diverse multimodal data sets. We have been talking about this, but we really don't have multimodal data sets. And to do that, you need a comprehensive, thrust towards collection of data from all over India by different groups. Now, who will do that? I don't know. I mean, there are government institutions who should do it, but I don't see too much of activity on that side. But the Digital India project by the Government of India, about which we have not discussed much, is trying to talk about it, but I'm not part of it, so I really don't know. But we do need, we are always using borrowed benchmarks. For everything, when you have a blood report, on the side they tell you if yours is between this and this, then you are normal. If it's between this and this, then you are, there is a risk factor. And then if it's, no, who decided these benchmarks? Is it applicable to Indian population? So that's something we need to do. And maybe, the academies or others would be able to create some kind of a forum, because they have fellows and they have people from all different areas, starting from basic biologists to clinicians to mathematicians to engineers, to have standing forums where such activities are discussed and, put forward for funding. Funding is something which is extremely important. There are regions of funding which is very unglamorous. You may be able to get funding for a particular, making a particular model, but then validation, deployment, and engineering and health economics again, where it reaches the level where everybody can afford is an area which needs to be pushed. And it's like from a research paper to a tool. I mean, that is what is the middle level, which is important. So I hope you did have a lot of things to think over, and all of you are as liked the session as we all did in organizing it. I thank the Academy for allowing us to do that, and everybody here for being here and listening to us. Thank you very much.
Thank you. So it's time for the
Hello, there's an announcement. We request all of you to return the name tags and the pouch back to the registration desk by end of the meeting. Thank you.